Hi guys, how are you? Welcome to Science Beyond Books, and I'm very happy that we are back with third episode of our Science and Research Simplified. We have successfully done two episodes so far, and there was overwhelming response from you guys. So come and join us, and the live session will start in another 4 5 minutes. Today's topic we are going to talk about is gut microbes. So one of the young scientists from USA, she's going to talk about gut microbes, and so once she joins after like few minutes we'll start the show. So before we start, there are a few things I wanted to tell you about the show. Is number one, if you are watching it on phone, I suggest you guys to use laptop or TV screen because we use presentations, we use images to explain topic. So it will be a lot more easier to understand things if you use your laptop or TV. But phone should also fine, but just because it's a regular show and you know that it comes every Saturday, so you just try to prefer using a bigger screen to understand the presentations or the talk we are going to do. So that's number one. The second thing is that first two talk we already did. So there was many questions which people asked, which we were not able to take during the session, during the live session. But what we did is after the live session was done. So once the live session is done, we upload these videos on our channel. So you can go to my channel and watch those videos like the first episode and second episode. So what we do is we try to answer those questions there in the comment section once it's uploaded. So if we could not take your question, like we cannot take all the questions, we'll try to take as many as possible. But if we could not take any of the questions, then if we could not take any of the questions, so what you can do is you can go back to our channel and watch the video again, and you will see your answer in the comment. So we'll try to do that. uh, the third thing is that if you don't understand English and if you have difficulty understanding English, what we will try to do is when we'll upload this video on YouTube after the live session is done, we'll try to put subtitle for English or some other language, local languages, so that you can understand it easily. So that's all for this thing. And one more thing is, like, as I mentioned you, that this is for mostly student who is in school or undergrad, to give them exposure of research. So try to reach as many as a student you can, so that they can get benefited from this show. Now, before we start today's show, just one last thing I want to tell you guys, that if give me one sec. Yep. So one last thing I want to tell you, that if you want to join us on the live screen, like when we'll be doing live screen, if you want to join us for any question you want to ask, so you can always ask your question in the comment section whenever you have doubt. We'll try to take those questions during the live session. But instead of asking the question in the comment section, if you want to come on screen and want to ask question directly to our speaker, you can also do that. But just let me know. I'll post a link in the comment section, so you can just copy that link and open in your laptop or mobile phone, and you should be able to connect to the live stream, and you can directly ask the question if you want. So that the link will be in the comment section. So I think it's already 8 p.m. in India, and it's 9:30 in USA. So we are going to start the today's talk. So let me invite today's speaker on the screen, and then we can start the talk. So hi Aathmaja, how are you?
Hi Ajit, I'm really good. How are you?
I'm very good. I'm very happy that you came to our show, and you are going to talk about your research experience and the topic you are doing your research in. So before, guys, we start today's talk, let me introduce our speaker. So she's Dr. Aathmaja. She did her bachelor from in microbiology from Itiraj College in Chennai, and she was topper in her BSc, and she also got Union Medal for the same. And then she did her master's from Madurai Kamaraj University, right? Master's in genomics. And that's how I know her, because we were in the same university and we had a lot of constructive discussions when we were, doing our master's there in my MKU. So she did her master's in my MKU, and then she also got IGSDH fellowship uh, to do her PhD from Cologne, which is in Germany. So she did her PhD from Germany, and her work was on the relationship between transcription and nucleoid associated proteins in E. coli. And right after your PhD, she did her short postdoc from the same lab, same university. and that time she was working on the CRISPR-Cas editing in E. coli, and that's the technique you know about, because that's the technique which got Nobel Prize this year. And I also made a short video on that how CRISPR-Cas works. And then finally she moved to USA from Germany to do her second postdoc, and she's currently doing her postdoc in Department of Chemistry and Biophysics from University of Michigan, and she's working on single cell microbiology using different analytical chemistry and microscopy. And she has a couple of international publications in many scientific journals. She has outstanding, career so far, and she is doing outstanding work in the field of microbiology. So that's Aathmaja for you guys.
Thanks a lot for your kind introduction, Ajit. I'm really happy to be in this platform today and discuss about my journey and about my research with your audience.
Yeah, thank you so much. So before we start today's talk, so today we're going to talk about gut microbes, right? And we're going to understand what is gut microbe, how how they influence our life, and what kind of research people are doing in this field. So we are going to talk about all those things during the talk. But before we start our talk, I just want to ask you this question: that how you came in research, what motivated you to become scientist, or how you end up becoming a researcher?
So in high school, I was a super average student. I was super bad at physics, math, and chemistry, and I was good only at biology. So if you would have come and told me that I will become researcher after some point in life, I would have laughed at you, because I always thought researchers are someone who are like born genius and they are always like intelligent and they know everything. That's how they could find new things in the world. So since I was good at biology, I did my bachelor's in microbiology while all my friends were doing engineering at that point. And when I was doing microbiology, the molecular biology classes really fascinated me, especially, how DNA replication happens, transcription happens, and so on. So I was really curious to understand how these mechanisms happen and how scientists are finding these biological mechanisms. But I had no idea about research that time. But I wanted to do research, like do PhD and do these kind of research to understand basic biology. So I applied for some integrated PhD programs across India. I didn't get anywhere. But
Hmm.
fortunately, I got masters in genomics in MKU, and that's where I really understood how research happens. And I was really fascinated by the microbial genetic classes where
So
with
because you talked about, yeah, MKU, for many people from North India, they are not much aware of this university. This is one of the best university we have came across when it comes to research. Like they do really hardcore research in MKU, basic biology, translational research. They are very good at research. I mean, for me also, that was the first time I got exposure of doing real research. Till my undergrad also, I was like same. Yeah. So continue.
Exactly. So
Yeah,
those microbial genetics classes were really fascinating. You know how Munawar sir, really
absolutely.
inspires us with simple and elegant genetic techniques, how people found out like about DNA replication,
Absolutely.
transcription, and so on. So that really fascinated me. So I did my master project in E. coli genetics and I wanted to continue the basic molecular biology research. So I did the same for my PhD. I did continue the PhD in molecular biology and genetics in E. coli. And now I am doing slightly different stuff in gut microbiota, and I'm trying to understand their interaction at the single cell level.
Yeah, that's very important topic you are working on, because um, uh, like most of the people understand and they believe that our when we talk about our physiology, we understand that okay, our kidney, our lungs, our heart, how it works, but we don't know there is a huge contribution of microbes or bacterium to keep us healthy, to keep us surviving, like because we cannot survive without them. So it's a mutual relationship between bacteria and uh, you know uh, us, like human body, and that's why it's very important to understand the relationship, so that we can get most of it and we don't get sick,
Exactly.
right?
Exactly. Yeah. I
Absolutely. Yeah. So let's start today's discussion. Atmaja, if you have any presentation or something, if you want to
yeah.
pull it up, I can put it on screen and then we
Sure.
can start.
Yeah. Just a second.
So guys, if meanwhile she's trying to get the PPT on, if you have any doubt, feel free to ask those questions in the comment section. If you have any suggestions, anything you want to share with us, feel free to give your comment and feedback. We'll be happy to take them.
Just a second, I'm trying to.
No worries, take your time. And meanwhile, I just want to ask my viewer that if you are watching this show for the first time, do a favor, subscribe to our channel, because we bring this regular show every Saturday for you guys, and we try to bring researchers from all different fields so that you get exposure of research in all different fields, whether being physics, chemistry, bio, from different fields. So do subscribe to our channel and stay tuned.
Just a second.
No worries, take your time.
I'm not able to find the application window.
So you don't have to, you can just go and open, yeah, so it's here. But you just open your PPT and that should be fine.
Okay, can you see it now?
Let me see. No, you have to, you don't have to share the window. You can just go and open the PPT. Share the
Okay.
presentation. Yeah.
Okay.
Yeah, yeah. Just share the presentation and then it should be there.
Is it there now?
You're still sharing the tab you are in. So what you
Okay.
can do is, yeah. So when you go and hit the share screen, you can
Yes.
choose
Yeah,
the.
when I go to the application window, okay, now I see it. It was not there before somehow. Now I can see it.
It might have gone for some
Yeah.
shopping.
Sorry for the delay,
No, it's
everyone.
absolutely fine. Let's bring this. No, because I think having some presentation or having some images helps young student to follow or understand. Because if we keep discussing, sometimes it's difficult for them to understand. Okay,
Exactly.
so before we start today's presentation, let me quickly ask you that. So what are the microbes? Why they're important for us? If you want to give some background about it, then we can talk more about the research in that field.
Sure. So microbes are nothing but bacteria, viruses, and fungi, which you've all heard about. And these are really small, tiny organisms which are unicellular, and you cannot see it by human eye. So eye can see something about 10 times less than 1 millimeter. So you these bacteria are 10 100 times smaller than that, so you basically need microscope to see these bacteria. And this bacteria, viruses, fungi, they are present everywhere in the world. It's there in the soil, it's there in water, it's there in all the animals, plants, insects, and it's there all over your body. So most of these bacteria, viruses, and fungi are good for us, and they help us live a better life. So if you want to eat your nice idlis and dosas, you cannot eat without the bacteria fermenting them, and you need bacteria to ferment your curd, cheese, and these bacteria helps us fix nitrogen in plants, and it provides nutrients for plants, and we eat plants so that we live. And cyanobacteria produces like 50% of the oxygen that we breathe. So uh, the oxygen you get is also from bacteria, and it gives enormous health benefits to the human beings as well. So we basically cannot live without these microbes. Hello.
So can you hear me now?
Yes, I can hear you. Did I cut off in between, or
No.
was it?
You are going fine.
Okay.
So yeah. So as you explain about the microbes and the importance of microbe, because we can see microbes are everywhere, as you mentioned, right? What are gut microbes? Do we have microbes in our gut, or what is gut?
Yeah, sure. So gut is basically anything from mouth to your large intestine, everywhere where the food passes. So we have about like 10 trillion bacteria in our body, and it's equivalent to the number of cells that is there in the body. So, and the largest number of bacteria in your body is in your gut. And there are about like 2,000 species in your gut with 20 million genes, and human beings ourselves have like 23,000 genes. So the number of microbial genes in your gut like outnumber the number of genes you have in yourself. So these gut microbes are really important for our health. It gives nutrition, it metabolizes complex carbohydrates for us, and it provides micronutrients for us. And it takes part in immunity, it enhances the adaptive and innate immune system, and it also takes part in the nervous system, in the development of central nervous system, and forming the gut-brain axis. So the gut feeling you are having is really real, and it is due to the bacteria that is there in the gut that's producing this feeling. There is always constant connection between your gut and the brain and the gut, vice versa. So if you have some problem in the gut, if you have like dysbiosis, if you lose good bacteria in the gut, if the bacteria takes up, you get a lot of diseases. You get like immune mediated diseases like inflammatory bowel disease, celiac disease, rheumatoid arthritis, metabolic diseases such as obesity, diabetes, and neuropsychiatric diseases like Alzheimer's, Parkinson's, and also infectious diseases. So it's really important to study these organisms and to keep the gut healthy, and so that we are healthy as a human being. So it's really important to study these gut microbes and what are the things that affect the gut microbes and so on.
Okay, so that's about the gut microbes, and yes, as you mentioned that gut microbes are important. We have like a number of bacteria residing in our gut, and they play very important role in our life. They has to be there so that we survive and we perform our normal physiology. So okay, so after that, if you want to tell a little bit more about your work, like what kind of work you are doing in this field, what are the questions being asked in this field, whether being related to your disease or what you want to understand in this field. If you want to tell something about that, that will be very interesting.
Sure, definitely. So most of the research that is related to microbiome or gut microbiome basically tells us what bacteria are there in the gut, how many are there, and what is missing between the healthy condition and the disease condition. So most of the studies are oriented towards the community. They just study the community, sequence all the microbes in your gut, and then check what is there and what is not there. What is missing in the field is to study what is the role of each bacteria and how one bacteria interact with other, and how this interaction forms this community. This nobody has understood yet. So it is important to study the role of each bacteria to know what it is really doing in our gut and which bacteria is important for us. So there are some keystone species in our gut, which are like primary degraders. The food you eat, they first degrade, and then they give the sugars to other bacteria, and the other bacteria survives because of this bacteria. So if this bacteria is gone in our gut, all the surrounding bacteria is also gone. So these bacteria are called keystone species, and they are very important for your gut health. So there are many things that affect your gut and the gut microbiome. One of the important thing that affects your gut is the food you eat, and the food basically contributes to the composition of the microbiome. The microbiome is different for people in Asia, and it's different for people in Europe and Africa and different parts of the world, because it really depends on the food you eat. And there are several research done on this, and people have found that if you eat more of resistant starch, fibers, legumes, and stuff, it is better for your health, because these starches and fibers are undigested by humans. So it enters the gut and it acts as food for microbes. So if microbes eat these food, they grow better, and you have higher number of microbes, so you have better health. So basically, what I am trying to understand is the role of one of the keystone species with one another bacteria in the resistant starch degradation. So these resistant starches are found in potatoes, corn, and other legumes. So what I am trying to understand is how a bacteria A is trying to basically degrade the resistant starches that is present in potato or corn, and what kind of sugars are released upon this degradation, and whether the bacteria B eats it and how it eats it and grows in the presence of other sugar, and what kind of products it releases in turn. So what I'm trying to really understand is the resistant starch degradation by one of the keystone species, and to know what kind of sugars are released, and how an A and B interact with each other in real time. Nobody has done this kind of studies as well. So I make use of microscopy techniques to really see how this bacteria grow in real time, so that it forms this kind of biofilm inside us. So yeah, as a part of my research, I also found interesting phenotype. Like one of these bacteria has a normal phenotype under high sugar conditions, and it has like elongated structure and at low sugar conditions. So this all I also published in microbiology. So these kind of small observations you can publish in your research, and that's how science is built. Like these kind of small observations are put together, and then you have uh,
Okay. Yep. So just let me ask you a very basic question, and then we'll move forward. So from
yeah,
where these bacteria are coming in our gut, like how it started? Do you have some idea, like how
yeah.
this evolve, or is it's a evolution of like several years that we got similar set of
Exactly. So
bacteria, or
this is a very interesting question. So what happens is inside the fetus, so when the child is there in the mother's womb, it's basically sterile, it has no bacteria. But as it passes through the birth canal during the birth, it acquires a lot of bacteria that is there in the mother's birth canal.
Mhm.
That's how a lot of bacteria goes into the stomach as well, and then it started colonizing. And uh, the human beings also get bacteria through breast milk, and then it kind of stabilizes at the age of 2. And then the composition basically is determined by the food you eat.
Mhm.
So it basically comes from your mother, and it is basically shaped by the food you eat in your lifetime. So if you eat a lot of fibers, legumes, nuts, and lot of things that has more fibers, then you have more gut bacteria, more diverse bacteria, you are healthy. And if you eat like a lot of simple sugars, like processed food, chips and stuff, you just have few types of bacteria that can eat that kind of food, and your gut is unhealthy, and you get a lot of diseases because of that.
Okay, so that brings one of the question. Chandrima Mukherjee has asked that which type of diet is better to maintain good bacteria. So I
Oh,
think that's what you're talking about. I think
yes.
she is your teacher. She
Yes,
mentioned somewhere.
she's my 12th standard biology teacher. I am here because of her now. So
Okay.
I'm really thankful to her.
Thank
Yeah,
ma'am. Yeah, for joining us. Yeah.
yeah, thank you, ma'am. So uh, the kind of food that we need to eat should have like lot of fibers, like I told, lot of legumes, whole grains. So anything that is not sugary and that is not easily digestible. So you should avoid processed food, and you should take complex carbohydrates. Like in Indian perspective, you can take different carbohydrates like ragi or barley, or if you take as many types of carbohydrates and fibers, it is better for your gut, because you will get those bacteria that will degrade these types of carbohydrates and fibers. So that's how you get diversity, and that's how you stay healthy. And lot of green vegetables as well, like fruits and vegetables, anything that contains fibers and these kind of
Yeah. So
complex stuff.
I think the understanding of what kind of food will help these bacteria to grow comes from the fact that if we know that what we eat will help bacteria to grow, if we understand that relationship, and that's something you are also working on, that
Exactly.
if you eat something which will be very helpful for you, but not be bacteria might not love it to survive. But there is something which not be very helpful for you, but it will help to bacteria to survive, and if bacteria survive, it will help in different ways for you to
Exactly.
be healthy. So in understanding that what part of the food is important for bacteria, you're not feeding yourself, you're also feeding those bacteria which is important for you. You're like you have your own you know uh, what a zoo or whatever. I don't know what to
Exactly.
call
It is
it.
a,
Growing
yeah, it is
inside.
as important as your heart, as
Yep.
your lungs, as your bones. So your gut microbes is as important as any other organ in
And
your body.
yeah, yep. And as you told, talk about it, right? I remember I have my own aquarium, and when I started, growing my aquarium, that's what people from where I bought it, they said that you cannot just put your fish into the water because they don't have their own microenvironment where they survive. You need to have some plants, some bacteria growing inside, which is important for them to feel like they're in pond or they're in their natural environment because fish never survive in just water. So that's why
Exactly.
we grow culture. We put bacterium and some those kind of microbes in water, and we stabilize water for like 2 weeks or something, so that there is a complete community is grown, and then
Hmm.
we put the fish inside. So it's some I can relate with that it's something like that
That's
in
true.
uh,
Yeah.
Yep. So let's move to the last part of this conversation. That okay. So we understood that what you're doing and what are the questions you are asking, and that's one of the very good question you're tackling, and one of the new kind of thing you're trying to do. But if my viewers, like there are many student who is watching the show, they want to know that how the lab look like. What kind of equipment you use, how you conduct these experiments. If you want to show something related to your lab, that will be helpful to visualize that where you work, what you do.
Yeah, sure. I can show uh, some of the places where I work. So yeah, this is the anaerobic chamber where I grow all the gut microbes. So microbes are completely anaerobes, so they cannot survive in the presence of oxygen. So our gut is
Just
also.
to, yeah, just to interrupt you for students. So if you want to explain what is aerobic and anaerobic bacteria, so that quickly they
So
understand.
yeah, aerobic bacteria are bacteria that needs oxygen to survive. So if you don't have oxygen, like it survives in normal room temperature and outside in the normal atmospheric oxygen conditions. And these anaerobes are bacteria that cannot survive in the presence of oxygen because it
Mhm.
is used to conditions like our large intestine has no oxygen at all in the place where it is living. So it is used to the condition without oxygen. So if you give oxygen to these bacteria, they just die. They cannot survive. There is some reactive oxygen species it produces and it kills the bacteria. So it cannot survive in the presence of oxygen. So I have to grow them inside these chambers. So these are like glove boxes. I should put my hand inside this and grow the bacteria and do all my experiments inside this chamber. And this is maintained at 37 degrees, which is our body's temperature. And I have all the conditions that is similar to our gut conditions inside this chamber. And this is how, if somebody is interested, this is how bacteria will look like if it's grown in the liquid culture, and this is what it looks like in solid media. So many different bacteria have different types of colonies, and this is like one such example I pulled out.
Can I just add something? So as
Sure.
viewers, you can see on the solid plate she is showing on the right side image. So every dot you see there is not one bacteria; it's colony of bacteria. So when we spread bacteria on these plates, all bacteria get separated from each other. Say for example, you have 200 bacteria in the culture. The culture you see on the left hand side. If you have 200 bacteria, and if you put them on the plate, what happens is all those 200 bacteria get separated from each other, and then they divide and divide and divide, and then they divide in such a number that they make a small colony. So every bacteria make one colony, right? Like when people spread from one place and they make a small villages. So it's like a small colony, and that colony has hundreds of bacteria. That's why you see those dots. Each dot is a colony of bacteria coming from one bacteria. Am I right, Atmaja?
Yes, yes, you're absolutely right. Yeah.
Okay, yep.
So yeah, these are just the bacteria that I grow, and I told you that I analyze a lot of sugars that is coming from this resistant starch. So this is one such machine that I use, HPIC, high performance ion chromatography, to separate the sugars and to identify which sugars are coming from where. And so our lab is also a microscopy intensive lab. So this is a normal inverted microscope I use. So under normal light, bacteria looks like this with phase contrast, and we also use a lot of lasers to see our bacteria. So we shoot the lasers inside the microscope, and you see that the bacteria can fluoresce if you have a fluorescent tag or fluorescent protein in your bacteria. And our lab is also specialized in single molecule imaging. Like you can track one protein in this bacteria throughout the cell where it is moving under life conditions. So we do a lot of these imaging stuff as well. Yeah.
Outstanding. So if you wanna, uh, that's a really nice presentation, and I think our viewer and many student who is watching the show might have learned about what people are doing in gut microbe research field. What are the questions they are asking? I have just one question, and then we'll try to take a couple of questions from the
Sure.
people have asked in the comment section. So you mentioned that you have bacterium A. What I understood is you are trying to understand that how these bacteria A,
Mhm.
degrade those starch, and what kind of product, sugar product they are making. So you're
Yeah.
trying to characterize those products first using these
Yes.
high technique, those analytical tools. You
Yeah.
use those analytical tools to look at that what are the metabolites
Yes.
when the bacteria break the starch.
Yeah.
Once you do that, then you mentioned that you also you are interested to understand the relationship of bacterium A and bacterium B. So
Yeah.
the product which is generated by bacteria A, which will
Mhm.
convert starch to something,
Yeah.
that something will be taken by bacteria B, and that will convert it to something else.
Yeah.
So you also want to understand the relationship between bacterium A and B, how
Yeah.
they talk to each other, or how they interact each other. If
Yeah.
so, how you do that? Is it something like quorum sensing or something related to
It
that?
is, so it's really simple. So the first thing I always do is grow the bacteria A in a tube and then remove the bacteria A. But all the metabolites it's produced, like all the sugars is in there in the tube. But I
Mhm.
just remove the bacteria and put bacteria B and see how it is growing, whether it is taking these sugars at all or it's just not liking these sugars and so on. And then using microscopy, I also grow them in live conditions and then see them grow, like how they are spatially organized. Like I told, these bacteria are smaller when they are in high glucose, and they are thinner when they are in low glucose. So when they are growing near these degraders, they look like normal bacteria, and they when they are far away from these degraders, they have these longer phenotypes. That's what will happen inside our gut as well. Like if we have regions that has low glucose, these bacteria will have a different phenotype, and if we have a region that has like high glucose, they will have a different morphology. So I am doing mostly in vitro studies, but at some point this can also be translated in vivo to human conditions. Yeah.
That's interesting. So the bacteria B you're talking about, right? I
Hmm.
know bacteria is defined that what bacteria you are looking for, because there are, as you mentioned, there are many bacteria, and you are interested in one particular bacteria.
Yeah.
So my question is that the bacteria B is already defined, or you are screening for bacteria B also?
It is already defined. Like there is a publication
Defined
about
means,
it.
yeah, defined means
I
you
know
are
what,
already in your mind, yeah.
I know what bacteria B is and what it does and whether it eats the sugar or not. Yeah, it is already defined, and it's also published before. People have done work on these as well. There are some studies that are studying, few bacteria these days, because that's what we want to know, which bacteria is really important for our gut, so that you can use that as a therapy. That if this bacteria is there, people are healthy, so people are less obese and so on. So people are really interested in understanding how each bacteria degrades sugars and how it affects the community. So if one key player you get, that you can use as a therapeutic purpose. So
So far, is there like few of the bacteria which is already? I mean, I'm not asking for the name of the bacteria, but is it already few key players defined already, or is still we're
yeah,
hunting for?
there are few key players, but
Still we
we
are exploring.
still we are exploring. Like healthy gut needs the community. So, like you need whole set of bacteria to make you healthy. So right now all the patients, like some people have inflammatory bowel disease, and some disorders like that, and people are treating them with fecal microbial transplant. So they take the feces from a healthy human being and replace it to the diseased human being, so that they can get the normal microbial community. So people are generally looking at the community till now, because the more bacteria you have, it is better for us. We still don't know all the key players in the community which are important for our gut health.
Okay, I have like hundreds of questions because it's very interesting topic, and for me also there are many questions. But I'll try to take some of the questions from audience first, so that
Sure.
their question get entertained. So there is one of the question. I'll just show it on the screen. You can see it. It says, "Is it better to stick to one healthy food plan all the time, or mix and try different type of good foods?" I
Uh,
mean, these are general questions. I think
I
it's,
know, I
yeah,
understand. So,
but if
uh,
you have any idea, it will be good to know.
from from research point of view, I cannot scientifically say that this food is good, like you should stick to this kind of food or something. But
Hmm.
in general, having different types of food is good for your body. Having different types of carbohydrates, different types of proteins, you vary it as much as possible, and you try to eat lot of like undigestible carbohydrates, like lot of fibers that you cannot digest by yourself. So uh, it is better to have as many different types of food as possible, instead of sticking to one diet plan. Yeah. And
Okay.
we as a scientist still do not know which is the good diet,
Yeah, we are
because
still working on, and
yeah.
the kind of study Aathmaja is doing, if we have more data, more information about, if we characterize most of the bacteria, understand the relationship, understand what they eat, what they produce, we will be in a situation to decide that, what kind of food you should eat, what kind of bacteria you need to maintain. But there is one similar question. Rahul Mani is asking how to stop or avoid losing good microbes. Is there any way, or we
This is
cannot
a
do it?
good question. Yeah, this is a good question. So there are several things that affects our gut. So one thing is food, I told about, and other thing is stress. So if you're stressed, there is evidence that all the good microbes are gone from your gut. So you have to try to be less stressful. And the third thing is exercise also stimulates keeping the good bacteria and in your gut, and avoid getting the bad ones in your gut. So that is one thing. And the fourth thing is antibiotics. Like if you take a lot of antibiotics, all your gut microbes are gone, and it really takes a lot of time to recuperate, to bring back all the good microbes. So these are the four things I would say. Like first is food, second is reducing the antibiotics, exercising, and reducing the stress levels.
Okay, so there is one more question. I think, I mean, you can choose to answer this because but you can see if you have the answer. So, is it the gut microbes that are the main reason for the obesity? Do you have any idea about it?
So yes, they are not the only reason for obesity, but there is a correlation. So if you take like 100 obese patients and 100 thin patients, and if you take the microbiome of these patients, there is correlation that in the obese patient these kind of microbes are there, and in thin patients these kind of microbes are there. So we cannot really say that the microbes are the only reason for obesity, but there is a correlation. There is also a study that there was a fecal microbial transplant for a diseased patient from a healthy person, and the healthy person was obese, but after taking the fecal transplant, the diseased person also became obese after a time point. So people are really trying to understand the role of microbes in obesity, but we still don't know which microbe is responsible for that, or is microbe the only reason for obesity. But there is a correlation for sure.
Okay, so now there are many more questions. Maybe probably we'll take one more question, but we'll take that question towards the end of the session. So before we take that last question, let me tell you something. Let me ask you something that, uh, for our viewers, she is a new mom, and so how you balance your life being, because now we have a lot of talk about women as a scientist. Now we have many women coming up, and they're doing outstanding job, because it's all about giving them opportunity. For like, for centuries we did not provide them enough opportunity to come and do outstanding research. And now if you see, we have like, the example is that recently the Nobel Prize we got, there are two only
Exactly.
ladies who got Nobel Prize. So now women are doing wonder in science. It's all about giving them opportunity. So how what do you think about that? How you are balancing your life as a researcher, being mom, and as a woman? Yeah.
Yeah, this is a good question. I think, it is really important to talk about these things, how researchers manage their life. It is not that we live in an isolated world where we don't have family or anything. We do live in a family system and we take care of everything. So, the one important thing is I plan everything. So, fortunately, my husband is also a researcher. So, he understands if I had a long day in lab and if I have experiments to do at night and so on. But one important thing I would say is to prioritize and plan your day. Like, you have blocks of time for doing reading, for doing experiments, for doing this. So, planning is really important. And as a new mom, I also had a lot of difficulties to come back to research after my maternity break because it was really difficult to separate the family life and the research life, especially in the times of COVID. So when I was at home working, it was difficult to stop myself to take care of my crying kid and so on. So but with time I understood how to separate these two, and I just go out of my home to my office to do work and then come back and then manage things at home. So it's mostly, women can have it all, I would say. So there are a lot of amazing women scientists. My boss is one, and my PhD boss was
Even
also
my
women.
my professor is, yeah, and she's
Exactly.
outstanding researcher. Absolutely,
Exactly.
she
So there
is
are a
known
lot
face
of
in cancer biology.
women,
Yeah.
female scientists around to get inspiration from. So it is not something impossible to do. You can, have a family life, do research, and do great things in life. So I don't think there is anything that will stop women in coming generation to,
So
do.
add something to that. So when I mean, I'm also doing research for many people who don't know that I'm also doing cancer research here in UNC. But anyways, so when my parents came to visit me for the first time in abroad, they were like, I see like a normal life. Like I had this feeling that scientists have one mouse running here, long hair, and
Long hair, and they don't do anything.
yeah, they
Exactly,
don't
they're
take
all in
shower.
the
Yeah,
lab. Yeah.
they don't take shower. They don't do anything. They're totally into lab. They sleep, eat, do everything in the lab. But that's not true. We are like as normal as any other people.
Any
We
other human
have
beings. And
yeah, we have
exactly.
like social life, and generally the way we do research is we have like like regular 9 to 5 duty. We go lab to a certain particular time, like 10 or something, or 9 or something, come back in the evening. But if we have some experiment to do, yeah, we stay overnight. Sometime we take day off also, because it's very flexible job. It's
Yeah.
not like some days, like you don't have to do anything at all. You sit and read book,
Exactly.
novel, sorry, your scientific journals and all. You do whatever you want. Sometime you have to work like anything for day, night, and maybe a couple of days. So it's like that. It gives you flexibility to plan your experiment, but it's like any other job or
That's
any other work we do. We are as normal as any other people.
that's
Yeah.
true. That's true.
I'll take one more question, and then probably I'll let you go, because I don't want to take a very long time from you. You already gave almost 1 hour. So I'll take one last question, and then probably we should be good. So if you could see the question on, it's Venkata Krishnan. He's asking that in a career perspective, can you please give me some insight on the future for this field? And I'll add something for this question, that if someone want to become a scientist or researcher in this field, what will be your suggestion or advice?
Yeah, so first thing is you have to read a lot. There are a lot of scientific articles that is coming about microbiome. This is a very new field actually. Only in the past 20 years we have started exploring about our microbiome, and only recently we know the connection between the microbiome and all those diseases, especially with the brain related diseases and so on. So we really know very very little about our microbiome, and there is a lot more to do, especially to know how this microbiome affects the diseases, how it causes the metabolic disorders, the infectious diseases, and the obesity and also the mental disorders, and how we can use this microbiome itself to treat these kind of diseases. So this is a very, very interesting topic to research on, and we know very little about it. So far what we know in this field is just a correlation. So disease person has this bacteria, and normal person has this bacteria. So this is what we generally do in the research, but people are now trying to understand what each bacteria does and how it interacts and so on. So as I told, there are about 2,500 species to study about, and people have studied one or two basically. So there is a lot more scope in this field to research on, and to know uh, more.
Yes. So thank you so much for this very, very interesting and important session. Before you go, I want to ask all my viewer that it's a regular show. We do it every Saturday, and we try to bring scientists from all different fields. As a scientific community, we can bring researcher to share their experience to talk about different research field. But it's your responsibility to spread this message to all the students. Whether being from undergrad or school going, we try to keep it simple so that the school going student also get exposure of research, and also undergrad, because as undergrad student we did not had any experience of research. And Atmaja will agree to that. So
Exactly, especially in India, like at least here I see undergrads doing research, publishing papers, and they have a fair idea what is going on in this research field. But back in India, I had no idea about research. I had only theoretical knowledge about
same
all
here.
the
Yeah.
process and everything. Only in masters I got to know about research and how
Yeah.
it is done. And
Yeah, absolutely. So if you are a teacher, I request you to share these videos and let them know that we have a show every Saturday 8 p.m. and we discuss different topics. It's not only biology. Like next week, we're going to talk about solar cells, solar plate. So one of my friend from Germany, he's going to talk about what kind of research is being conducted in solar cell or solar plates, and there are many interesting topics which has been lined up. So if you are a lecturer, professor, or teacher, let your student know and have a habit of watching these show regularly, so that they will be aware of what's happening in the field of like different fields when it comes to research. And thank you, Atmaja, for coming here. I hope you liked it.
yes,
And
I really enjoyed this conversation, and I think it's important for scientists as well to, go and give such talks to public, to know
general audience. Or
audience, to know what, to make them understand what we are doing. It's not some complex science, something that nobody can understand. It is quite simple.
and I'll appreciate that you kept it very simple also, and the presentation was very nice, very very clear and very interesting. So that's all for today, and thank you, Atmaja, for joining us. And
Thank you.
I wish you all the best for your research. And maybe and if we continue this program, maybe we can have another session with you, and we can talk more about your research in maybe more depth,
Sure.
more detail. Thank
Sure.
you for joining us. Have
Thank you
and
so much.
all the best for your research.
Thank you.
Yep. So, guys, that's all for today. If you are new on this channel, I request you to subscribe to my channel. We are doing two different kind of shows on our channel. One is I make science experiment videos. Unfortunately, those videos are in Hindi, so I make simple experiment for kid to inspire them to fall in love with research or experiments. So I do demonstrations and experiments. You can watch those videos on my channel if you are interested in. But apart from that, we are also doing this regular show where we bring scientists from different fields and we talk about different research topic. We call it Science and Research Simplified. There is one more show we're going to start soon. So my younger brother is a doctor in India, and we are going to start one more show on this channel, which will be about the medical field. So we're going to invite different doctors from different field, and we're going to talk about different disease. So if you have any question about those disease, you can ask those questions in advance, and we can discuss those questions from different doctors. Like we'll talk about the epilepsy, we'll talk about the diabetes, we'll talk about all those different diseases, and then we'll also talk about the different medical field. Like if you want to become, you want to do MD in pathology versus you want to do MD in any other field, how you can do, when you can do, what are the process to do, how you choose different stream in medical science. So there will be a parallel show coming from most probably from next week, where we'll be talking about everything related to medical, whether being admission or diseases or health related stuff. And we'll have this show also where we'll talk about research. So that's how we want to bring more awareness about diseases, health, and also the research. So stay tuned. Join us every Saturday, 8 p.m. On this channel. If you are new, do subscribe and share with your friends and family. That's all for today. Take care and take care of your health. Bye bye.